Aloha and welcome to our video on Mendel's discovery. The goals for today's video are to describe Mendel's work with the pea plants, we'll state the law of segregation, and we'll state the law of independent assortment, and then we'll practice a little bit with punnet squares. Let's begin our story of genetics with Gregor Mendel. Gregor Mendel was an Austrian monk in the mid-1800s, and he did a lot of work with breeding of pea plants. And what he did is he found a bunch of different traits, 7 of them he worked with, and he was able to find out how he could breed those plants together to manipulate those traits. He used a process called cross-pollination, which we can see down here, where he basically played the role of an insect and he would collect pollen from one plant and deliver it to the other plant. He's called the father of modern genetics because of what he discovered, and unfortunately what he did discover at the times was buried away for a while. His discoveries weren't found until after he had passed away, so he never saw the fruition of his work. He didn't see the net results of it, but I'm pretty sure that he understood that he was dealing with something that was pretty important. So as I mentioned, he had found 7 different traits that he had looked for. He looked at seed shape and he found that there was red and wrinkled ones. He found seed color, they were yellow and green. He found a seed coat, it could either be gray or white. The pod shape was either smooth or constricted. The pod color was green or yellow. The flower positions were either axial in the middle of the plant or terminal at the end. And then there were tall and short plants. And he was able to grow plants and breed the plants that he had these pure strands that would be either one of these conditions. So that means that if they bred, that's the only type of offspring they had. So he started off with his pea generation, which was the first time he would cross them. And if he took a trait, and we'll just take the tall and short for instance, he took a tall plant and a short plant and he crossbred them, that was his F1 generation. And they noticed that they were all going to be tall. So he got the ideas of dominance this way, saying that the tall phenotype, this tall one here, is going to be dominant to this short one here. And that's what he showed in this first generation. But he wondered what happened to the short, so when he crossed these first generations here and got the second generation, he noticed that he found most of the individuals were tall, but there were some short individuals that returned. And he noticed that that ratio was always in a 3-to-1 ratio. And this led him to do a lot of thinking, pondering, and that's where our ideas of genetics comes to mind. Now, Gregor Mendel came up with 2 laws that are important to genetics. The first one is this one here, the law of segregation. And the law of segregation, quite simply, we'll make it real easy. The lesson will go into it in a little more detail for you. But what the law says is that as I am a pea plant, and in this case a heterozygous pea plant, what I have is I have 2 chromosomes for a certain trait. During meiosis, these 2 chromosomes will then separate, and we're introduced to a term called an allele. And an allele is just that genetic trait. It can be various forms of the same thing. So if we're talking about color here, we can have a white flower allele and a purple flower allele, and those 2 alleles will separate into the gamete so that each gamete will only have one of the alleles. Okay, and that's important. As you recall, through meiosis, we send half of the data. So if I got one from mom and one from dad, when we make the gametes, we're either going to see mom's passed on or dad's passed on. Now, Mendel also came up with his idea of the law of independent assortment. And what he said is that alleles for different traits weren't linked to each other. And that's a key part. And we'll see later on that there are some traits that are linked, but he was fortunate. The 7 that he chose, they were on different chromosomes for the pea plant, which means they weren't tied together. So not all tall plants were going to have purple flowers. They mixed and matched and varied. And what he said is each of the alleles would then be its own trait passed on, and they weren't attached to each other. So in the example here, what you're going to see is we have an allele or a trait here, and we have one here. And because they're on different chromosomes, what's going to happen is we have the opportunity to make an entire huge selection of possible gametes from here. Now, Mendel started playing around and trying to figure these things out, and this is where we get to the idea of the punnet square. And you can see a completed one here, but what I wanted to do is I kind of wanted to show you how we filled these out. So I'm going to do it here as well. So the first thing we'll do is we'll start off with the father's genes. And it doesn't matter who is on which side. We just put our P generation, the parent generation is going to be there. So we're going to take a father who is a purebred tall pea plant. That means he's going to be a capital T, capital T. He's purebred because all he's going to give are the capital T's, so that his offspring are all going to have that capital T. So we start off by putting one of the alleles here and the other one here. Now the mother, in this case, you'll notice that she was a short plant. So she has two lowercase t's, and we'll put those down here. And let me change her color real quickly, and we'll go this way. Okay? Now, the order of filling in a Punnett square is, by standard, you take your capital letters first, and you fill down, then your capital letters, and you fill across, then your lowercase letters down, and then finally your lowercase letters across. So we'll fill it out. We have our capital letters coming down, so we're going to have a T here and a T here. You can see how this T is going to fill down this way, fill these boxes. We get this way, and there's no capital T's going this way. So we look for a lowercase t coming down. There's none, but we do have this lowercase t going across, and this is how we end up with our genotype of a capital T lowercase t. So now I have a heterozygous individual. Both of the parents were homozygous. They were the same. My offspring are going to be heterozygous. They have a dominant and a recessive allele, and we'll talk what that means a little bit later on. But physically, all of my plants are going to be tall. Okay, and that's kind of simply how we fill out a simple Punnett square. Now, what if we have more complex Punnett squares? What if we're looking at two traits instead of just one trait? Okay, so what we're going to do is we're going to breed two heterozygous individuals for two fictitious traits. And what we're going to do is we are going to say that both individuals are going to be heterozygous for two traits. So we're going to have a capital A and a lowercase a, and a capital B and a lowercase b, and we're going to cross two of these together. Okay, and what this will do is this will give us the opportunity for me to show you how we fill these out a little bit more. Okay, so let's start off and we'll figure out how we determine what we're going to place in the top here. Okay, so if we start off with our genetic condition of being a capital A, lowercase a capital B, lowercase b, then we have to go through the process of meiosis, and we can make four different traits, four different gametes from here. Okay, but we're going to see all the possible pairs. So I'm going to focus here on the first one here and realize that I can have a capital A or a lowercase a. Now, the capital A can be paired with either of these. So I can have a capital A, capital B, or a capital A, lowercase b. And my lowercase a can also be paired with a capital B, or I can go a lowercase and a lowercase, as we see here. So now I have my four possible genetic combinations that the gametes can have, and that's what we're going to place into our Punnett square. So I will start off with a capital A, capital B, capital A, lowercase b, lowercase a, capital B, and lowercase a, lowercase b. And we fill these in on both sides of our Punnett square. Okay? Then remember what we said. The first thing we're going to do is fill our capital letters going down. And we're going to start and we're going to do the A's first, then we'll do the B's. So I have capital A's here, so I'm going to change something real quick. Here we go. So I have a capital A, a capital A, a capital A, and you can see how we're just filling in the Punnett square as such. Okay, then we do our capital A's going horizontally, and we notice that they're in the first two rows, so we'll just do it this way. Okay, and now we fill in our lowercase a's. So we'll go down first. And notice I'm just following the pattern. I don't need to do it exactly. And then we'll come across finally. And you can see here we go. And now we are done with our A's. Now we move to the B's and we do the exact same thing. So capital B's going down, and we have these two columns. Oops. There we go. And then we do our capital B's going across. There we go. And then finally we do our lowercase b's going down. And you can guess the last step here as we do our final lowercase b's. And these will be going across. And now we've completely filled out our punnet square. Okay, and we can calculate out the ratios. We'll notice that we have one individual that's going to be capital A, capital A, capital B, capital B. The phenotypic part is what we generally tend to look at. And when we're looking at our phenotypic part, what we're looking at is we're going to say that all of these individuals, so I'm just going to green in here, okay, are going to be, okay, filled in so that what we notice is that we have 9 individuals that are going to have a capital A and a capital B, right? Then we can count out these ones. We have this one, and this one, and we have 3 individuals that are a capital A, lowercase b, and then we have these 3, which are going to be a lowercase a, capital B, and then we end up with our one special homozygous recessive that's going to be a lowercase a, lowercase b. And Mendel found this ratio, and we've found this ratio, and that's our 9 to 3 to 3 to 1 ratio. Okay, sorry this video is a little bit longer, but it's a lot more information. Good luck with the lessons, and as always, we'll see you in the next video.